Well, the next talk is by Chuck Lynch, who cannot be here today. Hopefully he is as quick as Wani. Fortunately, he's recorded.
Hi, my name is Chuck Lynch. I'm an assistant professor of neuroscience in the Department of Psychiatry at Weill Cornell Medicine, and today I'll talk about how we're using dense longitudinal sampling to study individuals with mood disorders, not by averaging data in large groups, but by tracking how frontostriatal limbic connectivity and symptom fluctuations relate to one another within individual subjects over long periods of time. This approach treats each participant as their own experiment, helping us identify reproducible and hopefully clinically meaningful brain symptom relationships at the level that targeted interventions would ultimately need to be delivered. Let me start by defining what we mean by dense longitudinal sampling. Unlike a traditional longitudinal design, which might include just a few time points per person, dense sampling focuses explicitly on within-subject analyses, treating each participant as an N-of-1 experiment. Individuals return for dozens of study visits typically. Neuroimaging data is often paired with clinical or behavioral assessments, which allows us to track individual trajectories and examine how brain and behavior co-evolve over time, something that a cross-sectional or even a sparsely sampled longitudinal design can't easily capture. This framework has been applied to study natural mood variation, which I'll focus on today, as well as a variety of experimental manipulations highlighted below, including limb restriction in the Cass 2020 paper, pharmacological effects like the psilocybin paper, and more recently, changes in brain structure occurring during pregnancy. Dense sampling has emerged as a powerful alternative to population-level approaches for understanding brain behavior associations. It aligns very nicely with what Kateri de Gratton, Steve Nelson, and Evan Gordon recently described as the high SNR path, a strategy for the field focused on maximizing signal-to-noise by deeply characterizing individual subjects and understanding the links between brain and behavior over time entirely within individuals. Now that I've introduced dense longitudinal sampling, I want to shift to a complementary concept, precision functional mapping, or PFM. PFM refers to the practice of delineating functional brain organization at the individual level, including the size, shape, and exact spatial location of functional circuits in each person's brain. This matters because individual network topology often diverges markedly from population-level maps, and those differences can be especially pronounced in clinical populations, like in individuals with depression or bipolar disorder, for example. If we rely too much on group-defined ROIs, we run the risk of either missing critical network nodes or blending signals from nearby but functionally distinct areas. In dense sampling designs, this kind of mislocalization can weaken or obscure true brain symptom relationships, and that's why we and others use PFM to define individualized network boundaries that are precise and tailored to each participant. Our recent work shows why individualized mapping is important, at least in the context of studying individuals with mood disorders. In both unipolar and bipolar depression, we often see that the salience network occupies a larger share of the cortical surface than in healthy individuals. One striking and fairly consistent difference is in the anterior cingulate cortex, where we see a rostral expansion of the salience network into subgenual cingulate. These kinds of shifts in network boundaries are exactly what group average maps tend to blur, and highlights why precision mapping is so critical when studying circuit function and how it relates to symptom dynamics within individuals. In our recent work, we found that connectivity between frontostriatal nodes of the salience network, specifically between the nucleus accumbens and anterior cingulate cortex, tracks changes in anhedonia severity over time. These findings come from 2 individuals with major depression who we followed for up to 18 months, collecting up to 124 15-minute resting state fMRI scans across 62 study visits, each of these visits being paired with detailed clinical assessments. This dense sampling design allowed us to model how symptom fluctuations, in this case anhedonia, related to circuit changes within each of these people, revealing a reproducible brain symptom relationship that held up both within and across these 2 participants. Now I want to share some new analyses that build on the data I just presented, but incorporating several key methodological refinements. The first involves temporal filtering of the clinical symptom time series. Specifically, we applied a low-pass filter to isolate slower fluctuations, changes that evolve over multiple weeks to months. This decision was guided in part by clinical intuition. Mood state switches in depression are typically thought to unfold gradually, not day to day. So we wanted to align our analyses moving forward with that time scale. Higher frequency variability, changes in the scale of days, may be more likely to reflect responses to situational stressors, reporting noise, or measurement error, rather than true changes in underlying brain state. And by filtering out this higher frequency variation, we hope we can focus more on slow, clinically meaningful dynamics that are aligned with the time scale that circuit plasticity and mood state transitions are thought to unfold. The second refinement relates to how we test the statistical significance of brain behavior relationships at the individual level. In our earlier work, we used circular rotations of the symptom time series to generate null distributions, but that approach in hindsight has several key limitations. For one, it has a limited number of unique permutations, and more importantly, it can preserve true lagged effects. For example, if brain connectivity at one time point predicts symptoms at a future time point. To address this, we implemented an autoregressive model that generates theoretically an unlimited amount of synthetic symptom time series. These synthetic datasets importantly retain the temporal autocorrelation and variance of the real data in each individual, but are statistically independent of the brain data. This gives us an unlimited set of realistic, behaviorally plausible null models and allows us to ask whether the effects we observe in the real data are stronger than would be expected by chance. Finally, we extended our analyses to examine other symptom dimensions, not just anhedonia. And to do this, we relied on partial correlations, which allowed us to isolate the unique variants in each symptom domain and link these more precisely to changes in discrete brain circuits. So, for example, we still found that weaker connectivity in the frontostriatal salience pathway, specifically between the nucleus accumbens and the anterior cingulate cortex, was associated with more severe anhedonia, but at the same time, stronger connectivity between the medial prefrontal cortex and hippocampus, both key nodes of the DMN, tracked increased feelings of guilt, low self-worth, and symptoms related to things like rumination. These opposing patterns across distinct circuits underscore the importance of symptom specificity, and importantly, both of these effects were replicated in a second densely sampled individual. This is probably my favorite finding in the entire talk. We next repeated the same symptom connectivity analyses I showed you on the previous slide, but this time we replaced the individualized network ROIs derived from precision functional mapping with a group-defined salience network. And the results were striking. The amount of variance we could explain in symptom fluctuations, the R-squared, dropped by nearly half. Now that's not because the group-defined ROIs are entirely wrong. They clearly still capture a negative correlation, but the effect is weaker, which we interpreted as being due to the group-defined ROIs not properly aligning with the true functional neuroanatomy in each of these individuals. I like to think of it this way. Using group-defined ROIs may be like trying to tailor a suit using average body measurements. It might fit okay overall, but chances are, in some people it will be too tight in some areas, too loose in others, and it's going to miss the unique contours that matter, importantly, for a good fit. You could think of the same principles applying to brain networks. If we want to explain symptom variation at the individual level, the precision of the ROI matters, and individualized maps give us a much better fit. In this slide, we can dig into this finding a bit more and better understand why the group-defined ROIs are explaining less variance. On the left, I'm showing again this group-level definition of the salience network in black, and overlaid around it are individual-specific salience network borders, defined using precision functional mapping. And you can already see that the individual map includes regions that the group map misses. On the right, we're showing you a heat map of the correlation between connectivity, and specifically between nodes of the salience network and the nucleus accumbens and the various vertices of the anterior cingulate cortex, with anhedonia scores over time. What's driving the strong effect is primarily this rostral expansion of the salience network into subgenual cortex, marked by that red arrow, a feature that's clearly present in the individualized map, but missing from the group-defined one. The yellow arrow shows that the group ROI does capture part of the signals, but it omits this key region marked by the red arrow, and that's what's contributing most to the effect, and that's why the explanatory power drops when we use the group definition instead of the individual specific one. Overall, I think this example nicely illustrates the cost of circuit mislocalization. It's not that the model is wrong, it's just not capturing all the signals of interest because the ROI in the group level case isn't aligning precisely enough with the unique spatial layout of the relevant circuits in this individual's brain. Now I want to share some really exciting preliminary data that extend this dense sampling approach to bipolar disorder. This data set comes from an adult male with bipolar 2, scanned 120 times over 60 study visits over a year and a half. What's striking is that we replicated the same circuit symptom relationships that we observed in the 2 unipolar depression cases. Specifically, during depressed-like states characterized by anhedonia, feelings of guilt, and rumination, we saw weakened connectivity between the nucleus accumbens and anterior cingulate, again, nodes of the salience network, and at the same time, increased connectivity between medial prefrontal cortex and the hippocampus, again, nodes of the default mode network. In contrast, during hypomanic-like states, primarily characterized by irritability and impulsivity in this individual, we observed increased connectivity between the posterior aspects of the middle frontal gyrus and the ventral pallidum, both of these being nodes of the cinguloopercular, or more recently termed action mode network. And when we take a step back and look across both the unipolar and bipolar cases I've talked about today, what emerges is a kind of working multi-circuit model of mood states switches, one in which fluctuations in discrete frontostriatal limbic circuits map onto different mood states characterized by different symptom profiles. And of course, this is still a very preliminary model, but we think it's an encouraging start. And over the next year, we hope to begin collecting new dense sampling data sets to test the generalizability and specificity of these effects across a larger number of densely sampled individuals. All right, I just want to conclude by emphasizing the idea that mood disorders are fundamentally episodic conditions, and our team has taken the approach to understanding the neuromechanisms that are driving mood state switches by adopting individual-focused longitudinal approaches, specifically this dense sampling strategy. And in this framework, each patient we treat as an N-of-1 experiment, and we think this gives us a really powerful lens on studying individual brain symptom relationships. And a key part of this approach, we believe, is the precision functional mapping component. By using individualized maps, we found that we can explain far more variance than when we rely on group average maps. And we think this underscores how important it is to correctly and accurately localize the relevant functional circuits at the individual level. And altogether, we hope that this approach will bring us closer to mechanistic, personalized models of symptom expression in mood disorders and hopefully set the stage for more targeted circuit-guided interventions. I want to close by thanking all the collaborators on this work, especially my mentor who made all this work possible, Connor Liston. All of the data that I present today was collected while I was doing my postdoc in his lab. I'm also deeply grateful to our longitudinal study participants who contributed a tremendous amount of time and effort across a very long period of time, and their dedication made this research possible. Looking ahead, we're actively building on these findings as we kind of build our program, including new dense sampling studies in unipolar and bipolar depression, and hopefully also some experiments using non-invasive brain stimulation to test causal links between brain circuit connectivity and mood symptom expression. So if you're interested in this kind of work, either as a prospective research assistant or a postdoc or even a collaborator, I'd love to hear from you. And thanks again for your time. I'm happy to take questions either now or over email. Thank you.
Let's once again thank all our speakers, and thank you all very much for coming.